which is a perfect segue for our next speaker. So I'm going to introduce Monika Fleshner from University of Colorado, and she's going to talk about prebiotics, probiotics, and stress. And Monika, turning over to you, and let's carry on.
All right, very good. Wonderful presentation that really did cover a lot of ground. And so everybody sort of strap on your learning hats, because I am a professor at the Department of Integrative Physiology at the University of Colorado in Boulder, and I teach undergraduate and graduate immunology. So I'm used to giving lectures. So I'm hoping to really teach everybody some more of the details at a more fine-grained level, and share with you some experimental data from my laboratory as well as others. So let's go ahead and get started. Okay, great. Can everybody see this? Is this in presentation mode? Not quite.
Not quite in presentation mode. There you go.
There we go. Are we good? So the work that I'm going to share with you today was supported by several different sources of funding, which I want to acknowledge. Mead Johnson Nutrition supplied the funding for the very early work, and the Office of Naval Research supplied the funding for the later work. So we're going to talk about prebiotics, probiotics, and a stress-robust phenotype. So the stress response, friend and foe. We usually think about the experience of stress, which I don't actually like that word because it's not really clearly defined. It can be a noun, it can be a verb, but we all understand that idea. However, if you want to understand the physiology of the stress response, it's actually incredibly critical for survival. We would not be here today without it. And there's been a great deal of work understanding how it is that when an organism recognizes that it is in danger, it needs to activate this powerful stress response. So for example, if you're the gazelle being chased by the lion, or the lion chasing the gazelle, both of them have activated this powerful stress response that functions in concert to mobilize energy. So things like your pupils dilate so you can see better, to run away or to chase. You change your saliva flow. Blood vessels in the skin constrict. The heart beats faster. You shut down digestion. No reason to digest if you're going to be somebody's meal. You can shunt blood to the brain, which is incredibly needy of glucose during these times, etc. And the underlying signals that create these physiological changes are also very well understood. Two particularly powerful axes that become activated is the hypothalamic pituitary adrenal axis, which begins with a particular set of signals in the brain, corticotropin releasing hormone that gets into the bloodstream that then ends up activating. We end up with adrenal corticotropin hormone, ACTH, which then releases cortisol in humans and corticosterone in other lower rodents. Of course, we also have this extremely fast-acting nervous system response too, which is part of what we feel. For example, if you were going through a stoplight and you realized that a car's coming toward you, you can feel that really fast response, and that really fast response is a neural response, the activation of the sympathetic nervous system. And less talked about, and I wish I had more time, is actually the impact that this positive and beneficial stress response has on the immune system. So the immune system itself becomes primed and ready to respond. So if you were wounded during this crisis, you would actually have a better chance of healing and avoiding infection. One of the first physicians to recognize the broadness of the stress response was Hans Selye. And so we talk about the good aspects of the stress response, but the good aspects are really when it's an acute event. That is, you turn on that powerful cascade, and then when the danger is gone, you shut it off. However, human beings are uniquely prone to taking natural responses and making them abnatural. or unnatural, I should say. So what are we going to do about the fact that we activate this response in a chronic fashion repeatedly? We activate this response to events in our lives that are uncontrollable. So there's no reason to keep activating the stress response. You can't run away from it. And that kind of repeated chronicity of the stress response, or if you were in a situation that was highly traumatic, those are the damaging types of stressors and stress responses. And so we already heard a beautiful introduction into the many things that have been studied that promote I call it a stress robust phenotype. So exercise, of course, we saw some work on that, and we've done a lot of work in laboratory studies and in preclinical models and shown how the brain changes with exercise. Mindfulness training, social interactions, adequate sleep, and of course, diet, which is what we've also heard a great deal about. So what is stress robustness? If you think about it, stress exposure to a stressor ends up breaking the ability to cope. And in some people, you can endure a great deal more stress before hitting the tipping point, I like to call it. Well, that's sort of stress resistance. Stress resilience is the ability to tip or have some of the bad experiences, but bounce back very quickly. So this stress robust concept is something that NSF, the National Science Foundation, is sort of pushing. So we've got these well-known interventions that we can control. And more recently, we're interested in beneficial microbes. So I was so thrilled to see that MVACI was already introduced. Mycobacterium vaccae is a really interesting positive microbe found in soil. And one of my colleagues has been studying that for quite a number of years. So what is it, the definition of probiotic as opposed to prebiotic? So a probiotic is defined as a live microorganism, which when administered in adequate amounts confers a health benefit on the host. So these probiotic or commensal bacteria are bacteria that we co-evolved with as human beings. And although there's a lot of focus on those microbes in the gut, they're actually important in many other places across the body. But we're going to focus today primarily on the microbes that are in the intestine. And so the gut microbiota is this study. This is this entire family or numbers of the entire population of these microbes that coexist in the GI tract or in the gut. And this carpet, if you will, if you were to look inside of this intestinal tube, there's this carpet of a layer of bacteria, and it does a lot of really good things. Excludes the pathogenic bacteria from interacting with your tissues, facilitates immune regulation, and also barrier regulation so that the contents within the intestine stay within the intestine and don't leak out into your sterile self. So it's been known for quite a long time that the brain can influence the gut. So during times of stress, one of the things that, especially if you have an inflammatory bowel condition, the experience of exposure to a stressor in your environment will oftentimes trigger a flare. We see that a lot. And that's the top-down signals from the brain interacting with the gut itself. But as I've alluded to earlier, more recently, we're starting to understand that's what's happening in the gut environment is also impacting the brain. So we've got both brain to gut and gut to brain. So the focus of the research I want to present today is trying to ask this fundamental question: can we optimize the health-promoting microbial ecology in the gut? So one approach to try to do that is to explore these families of substrates called gut microbial modulatory substrates. And so one of the features of the types of modulatory substances that I'm going to talk about are classified as prebiotics. And so by definition, prebiotics are substrates that are selectively utilized by host microorganisms, conferring health benefits. And the kind of things that have been studied and characterized and labeled as a prebiotic include human milk oligosaccharides, so complex sources of carbon, galacto-oligosaccharide, and polydextrose, actually. So I don't know why I'm not able to flip my slide here. There we go. And what's interesting about these kinds of substrates is that the microbes in your gut selectively utilize these for their nutrition. So you help to promote and support those types of good bacteria. So, a lot of people would prefer to ingest foods rich in probiotic bacteria, so the bacteria are already there. But there are some advantages to taking the other angle, and that is supporting what's already there and helping expand those populations. Some of those advantages include producing stable and reproducible changes in the gut microbiome, supporting and expanding existing probiotic species, and avoiding the challenges of live probiotic delivery and survival, increasing chances of adherent populations, that is, that these bacteria don't just flow right through the system, but rather that they set up a shop. They attach to the intestinal cell wall and they create their own niche. And minimizing risks of disrupting a functional ecology and having unforeseen consequences. So before I describe just a couple of experiments that we've done and others have done, how do you actually measure the gut microbiota? Well, one way you could do it is to by using animal models. You could actually go sacrifice the animal for the study, take out the intestine, and then actually sample the adherent bacteria. And those studies have been done. However, that requires a sacrifice of an individual animal at every time point if you want to look at changes across time. So a pseudo measure, sort of a surrogate measure, I should say, is measuring the fecal microbiome. And we also measure the products or the metabolites that those bacteria produce. So a lot of the work we're going to talk about today was the result of collaborations between Rob Knight and Antonio Gonzalez Pena at the University of California, San Diego. And the approaches that this team uses is you can either sequence something called 16S RNA sequencing or shotgun sequencing, which is a deeper sequence. So what we're talking about here is assessing the genetic code of the bacteria itself. And so when you have a fecal sample, you can measure remnants of the bacteria's genetic material to understand who was there. And then we can also assess the metabolites that are produced by the bacterium themselves by measuring them using a bunch of different rather complicated tools like mass spec. So one set of results that I want to share with you today is the result of taking shotgun sequencing on fecal samples that were collected from laboratory rats that were placed on very specific diets. So some of the advantage of doing preclinical work at this stage is we can control everything about these animals' environments. So we don't have to worry about a lot of the potential confounding factors that occur in other studies. So these animals are housed in the same environment. They get the same exposure to socially interact with other animals. They get the same ability to exercise, etc. The only thing different is that one set of animals were fed chow that were enriched in two very powerful prebiotic substrates, galacto-oligosaccharide and polydextrose. And after 11 weeks, we took fecal samples, and then we did this more deep genetic sequencing to try to identify what kinds of bacteria we actually have here. Now, just to remind you, if you have forgotten your bacterial taxonomy, bacteria are identified, and typically you can identify them at the phyla, class, order, family, genus, or species level. And then an even deeper analysis, you can get to strains. So by doing this analysis and using some really highly conservative statistical tools, we were able to show an increase. These are the animals that ate the prebiotic in specific species of bacteria. So one was Parabacteroides distisonus, and the other was this Clostridium leptum. So what you can see is that two specific species are identifiable and increase in animals who ate this prebiotic diet. In addition, one of the other points that people are interested in is trying to understand how these changes occur together. And it appears that they're related very closely. So what we're showing here are the relationship between the Clostridium leptum and the Parabacteroides distisonus. And you can see that they seem to co-regulate each other. Why are we so interested in these particular identified species? Because there's already been a lot of work done demonstrating their health benefits and the ability to label them as probiotic species. And so armed with this information, we've conducted a series of preclinical studies, and we have been able to demonstrate the change in the relative abundance, so the amount of this Parabacteroides distisonus with ingesting this diet. So once again, these are male laboratory rats. The red and control diet, the blue ate the diet that was enriched in galacto-oligosaccharide and polydextrose. And what you can see is as rapidly as 2 days, you get a big bloom in Parabacteroides. They're continuing to eat diet after 5 weeks, 11 weeks, we still have an elevation, and at 14 weeks, we have an elevation. Although the average of this group's increase here at 11 weeks is lower than the average that we saw at the beginning, a lot of that has to do with the tightening of the response. So we had a few animals that responded really fast, and over here, it's a more consistent response. So after continuing with a diet that's enriched in this prebiotic, we see this increase that is sustainable for 14 weeks. If we go the other direction, here's our Clostridium leptum, and here we can see the increase across time in this species as well. Now, one of the problems that that this field is suffering as a new and growing area of research is reproducibility. Reproducibility across laboratories from one side of the country to the other, reproducibility across species from rats to mice to humans. So I want to share with you some data from a collaborative project with a team at Northwestern and then my own laboratory. And these are the two scientists that ran these studies. This is Sam Bowers. He is the scientist at Northwestern that did this work. And this is Robert Thompson, who is a longtime research associate in my laboratory. I'll also just let you know that Sam here is also my son. So there's a little plug for that, which it was great fun getting to work with him. And then lastly, I'll share with you some data from Steve Lindeman at Purdue University, who's interested in similar questions. So once again, this is the effect on this Parabacteroides distisonus that I showed you from the University of Colorado at Boulder. And the results from Northwestern are remarkably similar. So here they have an actual 0-day time point. We didn't start collecting fecal samples until 2 days on diet. And so they all start out low, and then you see this beautiful bloom that tightens up across time. So across 2 different laboratories, we were able to replicate this robust finding. The approach that Steve Lindeman uses is he studies human fecal samples, and the D1, D2, and D3 are fecal samples from 3 different donors. And the approach he takes is to look at the effects of adding particular prebiotic substrates as carbon sources. So here's the polydextrose, which I was just talking to you about. And what you can see is you get a nice bloom here in column E of this same bacteria, Parabacteroidetes. And so now that we have a formulation that is really robust in terms of changing the gut microbiota in positive ways, we've gone on to conduct a long series of experiments in models, using animal models, and to demonstrate the broad effects that increasing the prebiotic species through the probiotic species through prebiotic diet has across the stress response. So it stabilizes the gut community structure. So this is alpha diversity. It constrains some of the bad parts of the stress response. It changes neural circuitry. It reduces the negative impacts of stress on emotional responses, and also protects sleep from stress-induced disruptions. I'm just going to show you the results of one experiment. So all of this work is published. I've listed the first authors on these publications and the years of these publications. So if you wanted to learn more, you could go to the literature and find that. These are all peer-reviewed publications. So how do you measure an emotional response in a laboratory animal? Well, you can't go up and put them on a couch. You have to use some behavioral tests, and the tests that we use involve identifying a behavior that's very specific. So freezing, as well as the shuttle box response behavior. And so freezing is just the lack of movement. It's an innate response. And the shuttle box escape, it requires the animal to run back and forth through this door. And if an animal is experiencing depressive-like symptoms, they fail to learn this simple task. And what we were able to show is that animals who were put on prebiotics were protected from a stress-induced reduction, this hyper-freezing response. So an increase in this test is a bad thing. This means that these animals are feeling fear and anxiety. Whereas over here, the animals who were on prebiotics before this test were protected. And this is that shuttle box escape response. So once again, a high level means they failed, they gave up, they showed a learned helplessness response, and animals put on the diet did not. And we study animals so we can get down into the neural circuitry, subserving it. And we know a lot about these two behaviors. And the circuitry that's important for this response is the serotonin circuit, and specifically changes in the dorsal raphe, a part of the brain which houses a large number of serotonergic neurons. And we know that what an extreme stressor does is takes the brake off of this response, and that's what contributes to these negative emotional effects. And so we hypothesized that maybe the reason that these prebiotics are protecting these animals from the emotional consequences of this stressor is because it's changing this neural circuitry. And that's indeed what we found. We saw an increase in these braking receptors. So the 5-HT1A receptors on the neurons in the dorsal raphe were increased. And so you break the response, and the animals are able to then resist the negative effect of that stressor. And so it looks like I'm running out of time. I will just let you know that we have been working very hard in trying to understand the specific signal, the specific microbe-associated signal that might be responsible for this effect. And one targeted zone that is showing promise are the bile acids. So these gut bacteria are critical for regulating bile acids that are released when we eat food. So primary bile acids are made in the liver from cholesterol. They're released into the intestine, and 95% are reabsorbed. And these gut microbiota modify the structure of these molecules. And we've done this work already, identifying some of these bile acids, and we've got a lot of promising evidence that these our two guys are ruminoclostridium, which is also Clostridium leptum, and Parabacteroidetes are associated with some of the positive changes. And so I'm not going to get into the details of this mechanism, but this is the approach where you're taking to try to understand more about how it is that something as simple as ingesting more prebiotics could change the way the brain functions. So the conclusion from my presentation is that prebiotics reproducibly impact gut microbial ecology, increase the relative abundance of probiotic species, and promote a stress robust phenotype. And with that, my laboratory says hello from Colorado. We're in Boulder. And if I have a minute, I'd take any questions if anybody has any. Thank you for your attention.
Monika, thank you very much. And I think it's great. And today is really the day where we really do bridge between sort of fundamental basic research and into applications of the research. And I guess as someone who's very familiar with sort of the microbiome, I would ask you, what was the original rationale for the combination polydextrose and the galacto-oligosaccharides?
Well, when I was invited by Mead Johnson Nutrition probably 10 years ago now to work on this, it was not something that was in my wheelhouse. They came to me, and the way those experiments were done is they actually came to us with several different combinations of substrates, and we were blind to what was in those diets. So they didn't tell us which combinations we were testing. And in the end, after testing a multitude of different substrates and different substrates, this is the combination that seemed to work the best together. You can test them singly as well, and that kind of work, we just didn't get as robust of changes. And in fact, one of the other diets that was labeled test diet produced even more robust impacts, especially immunologically, which I would love to circle back and revisit in the future. But that's the rationale, is that they've had tested them. They knew that they were molecules that were required bacteria to be broken down, to be utilized as carbon sources. So they were very targeted in that fashion. Yeah.
Yeah, that makes sense. And I mean, also we're seeing this manifest in a couple of the formulations in the marketplace with combined prebiotics, so that synergy of prebiotics with different mechanisms of action with single sort of approaches within the microbiota modulation.
Yeah.
So connecting the dots. So obviously, if this is going to have applications in product development and understanding of sort of food sources and everything else, HMO sources, obviously it's important to have that reproducibility. So maybe tie that reproducibility onto the next steps. I mean, if this is going to get commercialized in any way, the reproducibility is a foundation.
Absolutely. And one of the areas of research that needs to be addressed in the field is a better understanding of where you start, right? So what does your gut bacteria ecology look like before you start a prebiotic, and then trying to customize it for that individual. So if you have a community structure that, first of all, is very low in diversity in the first place, you've got some problems. Because it's hard to target and selectively feed some good bacteria if there's almost none present in the first place. So that's the kind of thing in the future that I see happening. The other thing is, like I said, we tested a multitude of different combinations on different aspects of the impacts of this traumatic stressor. And some of those combinations worked real well for some outcomes and not others. And so if you're going to take this combination of a prebiotic, because specifically you're really suffering from disturbed sleep as a consequence of some stress in your life, then we should be able to figure out the combination that targets sleep the best. And by understanding that circuit, I think that'll be in the future as well.
All right, Monika, thank you very much. I look forward to continuing this conversation and an ongoing dialogue with you, your team, and your work. So thank you.
Great. Thanks so much, Len, for the invitation.